stop me anytime. First of all, can you hear me?
Yes, we can hear you.
Okay, and you can see me, you can see the sharing, everything, right?
We can see you on your screen.
Okay, good. Okay, just wanted to let you know that there are 5 materials are uploaded on Moodle for both of you to year 2 and year 3. The lecture 1 that we finished, lecture 2 what we are doing right now, then a short lab book. Then there is some keyword list, and also all the Olivia slides that I usually use is also if you want to use Olivia Virtual Microscope. So the second lecture, what we are doing today is talking about basically the epidermal appendages of the skin or epidermal derivatives. What does that mean? That these structures invaginate they are invaginations of the epidermis, and they are sitting in the dermis, and they are all secreting something. It's really interesting. So we're gonna go through the structure, function, location, some clinical application of these 5 structures. The hair follicle that is secreting hair, nail bed secreting nail plate, sebaceous gland secreting oil or sebum, pseudoporous gland that secretes sudor or sweat, and mammary gland secreting milk. And that one we are going to skip because that usually we cover in the reproductive system. So let's look at the next slide, which is kind of like a principal slide, what I call the push-up secretion. So what I taught you in the epidermis in lecture 1, that the mitosis occurs in the stratum basale, and then the cells are migrating upward, as I wrote it here, upward migration. This principle is actually applied to all of these appendages, including cell death and apoptosis. So in the epidermis, what we saw was that the epidermal cells were choked to death by soft keratin, and we ended up with dead corneocyte squams. Looks like a fish scale. Then in the hair, actually, situation really is also similar. The hair follicle is actually the extension of the epidermis, and these are the matrix cell, which is kind of like the, as I wrote here for you, red dots, kind of like the stratum basale in the epidermis. And this is where the mitosis occurs, and the hair is just pushed upward. So it's migrating upward until your hair is sticking out from your skin. And this type of keratin is actually another kind, hard keratin. If you look at on the other side, the sebaceous gland, the sebaceous gland, the same principle applies. At the edge of the sebaceous gland is the stratum basale. That's where the mitosis occurs, and the cells are migrating and pushed up toward the lumen of the hair follicle. The difference is that in the epidermis and the hair, the end product was keratin. Here, the end product is actually a mixture of lipid that is going to explode. And these cells are called sebocytes, and basically moisturizing the hair and the skin. And I put here one question that if you like to answer, that's fine. If I don't, why are these called sebocytes, not adipocytes? Anybody want to answer? So sebocytes are basically originating from the epidermis. So these are basically epithelial cells. The adipocytes, what you learned previously, you know, these white diamond ring-shaped cells are actually connective tissue cells. Okay, and finally, let's look at the nail plate. So this is my favorite orange finger slide where you can see the fingerprints on one side. Here is the deep blue dermis, and here's where the nail is growing. The nail is growing, and these are the matrix cells where the actual proliferation and mitosis occurs. And here the nail, again, another kind of hard keratin is pushed out as it's growing. Okay, so let's look at the next slide. Okay, so this slide is a little bit of review for you about the hair follicles, because hair follicles are also invagination of the epidermis of the keratinized stratified squamous epithelium that we covered yesterday. And what I wanted to just briefly cover for you, that these are let's go from bottom up, the way it is growing. So this is the derma papilla here, where the capillaries are, and where this keeps this whole structure alive, very similar to the epidermis. Then around this is technically the stratum basale, that we, at this point, we're gonna call hair matrix cells. Now you can see that the dark guy is the actual hair, and the hair has a part which is called shaft sticking out. The root is inside the dermis, and the hair bulb is deep in the dermis. Okay, now the actual parts of the hair follicle is, which is pink on the slide, pink, left and right. This is the infundibulum, which is above the sebaceous gland. There is a narrowing isthmus below the sebaceous gland, and there's a bulge between the sebaceous gland and the arrectopili muscle. And if you remember on lecture 1, this is a territory for a reservoir of stem cells. And we already covered the hair bulb area here. Looks like an onion. Some people call it the hair onion, and where the matrix cells are sitting, and the capillary is bringing in the supply. Now, what kind of nerve endings are sitting here? We're gonna do this again, but you're gonna have it's not on the slide, but you have a hair nerve plexus hugging which is actually a free nerve ending, hugging the hair follicle. And then you're gonna have a Merkel touch dome made out of 150 Merkel cells hugging the hair follicle. There are also sympathetic nerves here, motor nerves. Who needs sympathetic nerves? The arrectopili smooth muscle, blood vessels, smooth muscle in the tunica media, and sweat gland secretion. And you remember the arrectopili smooth muscle is the goosebump muscle. Just one word I would like to say it here for you, that I wrote only sympathetic nerves here because there are no parasympathetic nerves in the skin. The vagus nerve never enters the skin. So that's why the sweat gland eventually becomes like sympathetic nerve with cholinergic, which is very unusual. Okay, the 3 layers of a hair shaft is, please observe that I start with the cortex, which is basically keratinized epidermal cells and has melanocytes here for the color. That's basically the yellow. Then in the center, the orange is the medullary layer, or the medulla of the hair shaft. And this appears only in thick skin. And the outside, where we usually put the conditioner or the hair mask, is called the cuticles, made out of flat cells. Now, the parts of the nails is not meant to be memorized, like eponychium and hyponychium. What I wanted to show you was here is that this is really the place of the kind of like the stratum basale, where the skin is, not the skin, the nail is growing. Now, I wanted to also introduce you to the growth of hair as it goes through 3 phases continuously in cycles. So the anagen is when the hair is actively growing and getting thicker, getting stronger. The catagen, which is written here, this is where the hair follicle and the hair start degenerating. Okay, and the telogen is the resting phase, and this is where the hair follicle is totally totally degenerated. So this is in the process of degeneration. This is degenerating. And finally, this is where the new baby hair start growing. And this is what we're going to call early anaging. So when the old hair falls out, the new hair starts growing. Okay, so how does this work from our different locations on our body? The scalp hair, right, where we have the long hair, spends a lot of time on anagen cycle for about 2 years or sometimes even more. And therefore, it's become really long. The pubic symphysis, the trunk, the eyelash, the eyebrow, wherever we have shorter, mustache, the anagen cycle and the telog they're very short. So therefore, these hairs never grow long. Okay, so this is a kind of review about the different kind of secretions on the different types of glands, because we are going to cover 3 glands. So I hope you still remember that the most common way of secretion of these exocrine glands in the skin is really exocytosis. And the big word what people also use is eccrine or merocrine, and this is associated with the, the sweat glands and the regular sweat gland and the apocrine sweat gland. Excuse me. Here's the exocytotic vesicles that I like to also call it. The cell is spitting out the secretion, the sweat or sudor, and it reminds me of a geyser. The second option of secretion is the apocrine. It gives it away when you look at apo, which is the big word or the abbreviation of apex or the tip. Crine is always secretion. And what happens that the epithelial cells, the top of the epithelial cell jump off, or I call it suicide, like in mammary glands. The third kind is the holocrine secretion, when the entire epithelial cell of the gland explodes and becomes the secretion. And that's would be the best example of the sebaceous gland. And you remember we just did that in lecture 1, that the melanocytes, between the melanocytes and the keratinocytes, the cytokine secretion is a new kind of secretion exists when the keratinocytes are phagocyting, eating off a piece of the process of the melanocytes that are filled with melanosomes, a type of endocytosis, actually, which it reminds me of cannibalism. When somebody is biting to somebody else, that's cannibalism comes to my mind. Okay, so this was just like introductory slides for you. I would like to show you now 2 sections of this lecture. The first part is about the glands, the sweat glands and the sebaceous gland. And then the second part of the lecture is about the sensory receptors. So observe that we are starting with the sebaceous gland or the oil gland. And I put here for you as a reminder of the classification of the glands. On the top, from A to G, are the simple glands. Some of them are really simple, some of them is a little bit branched, like here. And on the second row, you can see the complex big glands. So where is the sebaceous gland? Where does it belong? It belongs to the FNG, which is basically the branch alveolar gland. Branched because you can see it's branching off left and right, and it's alveolar because the acinus or the secretory unit looks like a little bowl, little alveolus. Or you can also say it looks like a piece of grape. So the whole thing looks like a cluster of grapes. Now, if you look at in this other image, the sebaceous gland, you will see that this is, let's say, where did they, oh, it's not labeled. Okay, well, I'm going to show you. This is the sebaceous gland hugging the hair follicle right here. And what is also very nice on this picture that you can see two types of pores on the surface of thin skin. These are the sweat droplets coming through a sweat pore. And the sebum or oil is going to enter the hair follicle, and through the hair follicle, and there's a little pore here too where the hair comes out and the oil comes out, and guess what, who else comes out? The apocrine gland, sweat gland secretion too at the same time. So 3 guys are exiting here. Okay, let's look at the, so this is the big slide of the structure, function, location of the sebaceous gland. And structurally, as we just said, it's a branched alveolar glands, and actually several glands are hugging one hair follicle that are divided into lobules. You can see the lobules very nicely on this image. Now, this is a very unique gland. Most glands are made out of usually simple cuboid epithelium, but this gland does not look simple cuboid epithelium whatsoever. It looks stratified, and that's exactly what it is. The stratified squamous epithelium of the epidermis is modified in this gland. You see what you see? Round epidermal cell filled with lipid droplets, and you know lipid droplets wash out during processing, and that's why all of these guys are really white, like Snow White, and that's why this gland, you can't miss it. It's very easy to identify. Okay, so these specialized epidermal cells have a new name, the sebocytes, and there are desmosomes between them. So basically, after after exiting the acinus, the sebum or the oil is going to enter a little duct, little bit of little duct here, and ultimately the hair follicle become the final duct for delivering the sebum. Okay, exactly. So what is the function of this guy, of the sebum? It is a mixture of lipid, and we just did that, and it has moisturizing effect, it has antibacterial and waterproofing effect on our skin. Usually, the sebaceous gland is sitting in the pilosebaceous unit that we're going to cover in a minute. Pilo refers to the arrectopili muscle. Sebaceous refer, of course, the gland. And the third part is the hair follicle. There are exceptions for this rule. Sebaceous gland can also exist without hair follicle at the nipple, vulva, eyelids, and oral mucosa. So we already covered this actually. What kind of mode of secretion can occur here in about 8 days? The holocrine. It's also another program, cell death. And you remember I called this explosion earlier. So let's do it again. The mitosis occurs here in the stratum basale at the edge. Okay, and this is where the cells are having beautiful, normal, round, healthy nuclei. And then comes the migration toward the duct and toward the hair follicle, and the cells start dying. And eventually the cells rupture and the oil flows into the duct and eventually to the surface of the skin. The control of this gland is related only to sex hormones. And we all know that these glands, this sebaceous gland, wake up at puberty when the sex hormones start appearing. as far as I know, and the histology, according to the histology book, there is no nerve innervation here. Okay, I think we covered everything. And now this is basically our slide, which is kind of like, you know, combining with lab. So this is our slide in the histology lab. This is the epidermis, and then the pink is the dermis, and then you can see the white here is the hypodermis fat. Now, I'm introducing you to the pilosebaceous unit that is mainly sitting in the dermis. As I told you, these guys all epidermal invagination into the deeper dermis. So let's look at it. So this is basically the beginning of the hair follicle. Then the cut didn't go through here, but basically this is the hair follicle. And this is the extension of the epidermis. Then comes, you see these white glands. These are the sebaceous glands, white and filled with fat, hugging the hair follicle. Inside the hair follicle, you can see the hair. It looks like a blonde somebody. And then, on this slide, you can see beautifully the arrectopili smooth muscle, as it is on one side is attached to the hair follicle. The other side is actually attached in the dermis. On the right side, what you see is actually zoom in. So let's look at the sebaceous gland again. I zoomed in a little bit closer. This is where the mitosis occurs, happy nuclei, migration, holocrine explosion, secretion occurs. But before it occurs, you can see that the nuclei are not happy. They are already fragmenting and distorted and irregular shape. This is what we call pycnotic nuclei. Okay, then this is just a reminder how a smooth muscle tissue looks like with the spindle-shaped cells. If you would zoom into the arrectopili smooth muscle. Now, this is the hair follicle in transverse section, and 3 is basically the hair, and 4 is the different layers of the hair follicle. And the next slide is going to show you that this image is when the hair or the hair follicle is still very young, so to speak, as it's, you know, growing from the bottom up. This section is actually below the sebaceous gland, like here. So this section below sebaceous gland, and you see it's one of the most beautiful histological image ever. But if you go at the level of the sebaceous gland or further above, that's when the hair follicle really start looking like the actual epidermis in the surface, this purple stratified squamous and purple stratified squamous. And now you can see the lobules of the sebaceous gland are hugging the hair follicle. Okay, so cut at the sebaceous gland or above, it really looks identical with the epidermis, but below, it is still in a developing and maturing stage. So pimples or acne vulgaris or simple acne. So how does this develop? I think we all suffered from this at one point. A disease of the sebaceous gland and the hair follicle. So remember, it starts at puberty when these guys wake up. And as the sex hormones appear, a very proliferating, increased mitosis occurs. So lots of mitosis occurs. So therefore, lots of dead skin cells will start clogging the duct of the gland and the hair follicle. So but the oil synthesis never stops. So the dead cells are going to starts building up inside the hair follicle. And the moment there are dead cells and backup of any kind of secretion or fluid, bacteria is very happy. Breakfast, lunch, and dinner, they looking at it as their, food. And as the bacteria start growing, it's gonna develop an infection and inflammation. And we often, everyday language, everyday life, we call this whiteheads or blackheads. But as long as they are white and blackheads is not that problem. The problem is when the bacteria is going to start secreting pus that is not very attractive, and you can see here. Okay, so we are done with the sebaceous gland, and we're going to look at right now the two sweat glands, the eccrine, which is the regular everywhere, and the apocrine sweat gland, which is the at the the stinky sweat gland in a couple of locations. Of course, they're exocrine glands, and let's see where they belong. They belong to the group of B, simple coiled tubular gland, or I like to call it a coiled snake. So when you look at this guy under the microscope, every coiled tube, when you cut it, you're going to end up with circles. Okay, so let's identify on this image. The smaller coiled snake is the regular sweat gland. This is the duct traveling through the entire dermis, and this is the pore where the sweat comes out and cools up the body by evaporation. Now let's look at the other sweat gland. This is the apocrine sweat gland. This coiled snake is much bigger. And look what's happening. The secretion is not going directly to the surface in a pore, but it's gonna go merge into the hair follicle above the sebaceous gland. So it's a unique situation. Okay, so how does this look like on the slide, guys? So this image is about your regular sweat gland. You can see here a thick epithelium, stratified squamous, thick keratin. This pink thing is just keratin, so very thick epidermis. Let's go, and again, this pink thing is the dermis, and then the hypodermis doesn't show very clearly here. Maybe this is the hypodermis. Anyway, on this, we need to zoom in, but I cannot zoom in. So I made a zoom sign here for you, showing you the little circles, what I just mentioned. These are individual little 1, 2, 3, 4 sweat gland ocini sitting next to each other. And there is more here and here and here and here. I cannot label every single one of them. Okay, so these are the ocini that the coiled snakes that actually secreting the sweat, and I magnified one of them on the right side. And this sweat gland looks exactly the way usually a gland looks like, that you will see in the ocini, simple cuboid columnar kind of epithelium. Okay, number one. So if you look at it carefully, this one, I'm not sure what this is, but this guy, let's start here. This guy, continue this way and then go this. I'm showing you the coiled snake, and this way and this way and this way and this way. Up till here, it's all the ocini. Observe that the cytoplasma is very light pink. Number one. Number two, look that on the outer edge, there's this pink kind of ribbon here and here that I labeled with number 2. Those are myoepithelial cells that are helping the secretion to be pushed forward in the coiled snake. And then look at the scenery change. Do you see these guys are much darker and they have double layer of nuclei? So this is the double cuboid epithelium that is the typical for a duct. So if this is the coiled snake, the duct begins here and continues here. So you can see the ocinus and the beginning of the duct here. So if you go back to the other slide, you can see that this is now the duct, one duct, but we don't have it. So let's go here. So let's say this duct is maybe the duct of this gland and traveling through the entire dermis, and eventually it has to travel through the entire epidermis, and it has this spirally duct structure, what we call acrosyringium, and ends up in the pore. Okay, so everything is what I just said is written here. Simple columnar epithelium or simple cuboid, myoepithelial cells, duct in the dermis here, a little bit here, then traveling through the entire dermis, through the epidermis, acrosyringeal pore. So everything is written here. It's very simple. What is really interesting about this slide is that you also have Pacinian corpuscle here. And if we would be able to zoom in, this would be actually after the next layer would be skeletal muscle. Okay, this is a zoom into the duct of the sweat gland. So you can see a little bit closer what I just told you a minute ago, that there's double cuboid layer. It doesn't look very cuboid on this side, but you can see two layers of nuclei. And the black arrow showing you to this dark pink line, which is the microvilli. So in a longitudinal view. And this one is, well, kind of like an overview. Again, you can see better here the double cuboid because the nuclei are in two layers and the nuclei are round. And again, the pink line is the microvilli. Okay, so what is the microvilli doing in these ducts? They are reabsorbing sodium from the sweat. We all know that sweat is salty, but we also know that sodium is very important in many physiological processes. So the body is trying to preserve, to save some of the salt from the sweat. So the isotonic sweat, that usually comes out, becomes hypotonic after the reabsorption of the sodium. Now this slide is actually showing you the comparison of the regular ordinary eccrine sweat gland that I just showed you. And this would be the typical look of an, also in the dermis, of the other one, the stinky sweat gland or apocrine sweat gland. Okay, so let's compare them with each other. So the eccrine sweat gland is always all over the body. You know that sweating is everywhere. And these sweat glands are not connected to any kind of hair follicle. And we know that the duct goes to the pore. What does the apocrine gland is doing at the same time? It's not all over, only a few places, the stinky places in our body. And they are usually associated with hair follicle, as I showed you earlier. And the duct goes into the hair follicle or the secretion above the sebaceous gland that I showed you earlier. Now, how does the structure? Both of them are coiled snake, and they appear like circles, right? However, the coiled snake of the eccrine sweat gland is smaller. Smaller snake, smaller circles, smaller lumen. And three types of cells are sitting in these acini. How about in the apocrine sweat glands? This is usually larger, even though in this image it's hard to see it like that, but I will show you another one. The lumen is larger, you can see, to comparison. And what is really interesting, that it has like this bleb, like protrusions into the lumen. But again, observe, both of them is famous first, made out of simple cuboid or kind of simple columnar epithelium, which is the typical. And this is where all the exocytosis is going to occur, where the sweat is going to go into the lumen. Okay, two types of secretion shows up here. So this guy can do watery secretion, but it also can do a little bit of protein secretion into the sweat. These glands are active forever. They better be, because thermoregulation is very important. How about this guy, the apocrine stinky sweat gland? It has one type of secretion very viscous, high protein, lunch for bacteria kind of secretion. That's why they become stinky very easily. Bacteria loves to munch on proteins. And these guys, as we covered earlier, maybe we didn't. Anyway, they also wake up at puberty by the sex hormones. Oh yeah, because what we did was the sebaceous glands also woke up at puberty by sex hormones. So they wake up at the same time. Okay. So if you like tables, everything is written here for you, okay? And one more thing, I wanted to make sure that you, again, these are your epithelial cells, usually simple cuboid, simple columnar. Outside is the myoepithelial cells, and the same here. Look, these are the more like simple columnar epithelial cells, and the ribbon around the actinus are the myoepithelial cells. Okay? So this covers everything. If you love tables, then here we go. Okay. All right, so now I'm gonna show you sweat glands with a special stain, my favorite orange skin slide. More precisely, it's really your fingertip showing the nails on the top. Okay, like this, right? Like this. Okay, so this is the nail. These are the fingerprints. And you can see that the epidermis is all stained with beautiful orange color. And then in the dermis, the collagen fibers are blue. Let's jump over here. These are the orange acini of the sweat glands. And if you jump back here, you can see sweat glands are like everywhere. The collagen is very thick type 1 collagen. The myoepithelial cells don't show very clearly on this slide. The fibroblasts are beautiful, pink cells. And on the side, you can also see some adipocytes, so white adipocyte cells. Okay, so now this image is comparing for you the orange thick skin and the pink thick skin. So here, what you can really nicely see the fingerprints up and down, up and down. And what is really nice that this is the only slide that I can really show you the stratum lucidum of the thick skin. If you look at carefully, this would be this whole dark orange would be all epidermis with the 4 layers. Then this would be the stratum lucidum. And then on the top of the stratum lucidum, this is the stratum corneum. So this different shades of orange gives it away. So 4 layers of epidermis, stratum lucidum, and stratum corneum. And you can also see very nicely, these are the acini of the sweat glands. You see the little circles, and the ducts are going through. It's not easy to see the ducts here, but eventually they become acrosyringeum in the stratum corneum. And we already did this earlier. This is the pink skin. These are the fingerprints. This is the acrosyringeum spiral, where the sweat is gonna come into a pore. And I think we did everything. Okay, so thermoregulation is very important. This is the picture of hyperhidrosis, when somebody is secreting too much sudor. And hypohidrosis refers to the secretion of sudor when it's too low. And if the thermoregulation is not working, it can be really deadly. For example, anhidrosis, if let's say somebody was born with a rare inherited disease, have no sweat glands, they could die in heat stroke. Okay? So here is like, I know that physiology people spend a lot of time on this, but I also wanted to summarize for you how the skin contributes to thermoregulation. You know, the control is in the hypothalamus. The cooling off happens by evaporation of the sweat, or the capillaries go through vasodilation. And in terms of keeping you warm, You remember the goosebumps by the arrectopili muscle? Muscle contraction creates ATP, and the ATP becomes kinetic energy and heat energy. Okay, vasoconstriction also preserve heat, and of course, our favorite fat in the hypodermis. Okay, I just wanted to mention it to you because I thought it's really interesting that most animals have no sweat glands, if you didn't know, except the horsey. Horsey has a lot of sweat glands, but cold-blooded animals, pigs, ducks, rodents, and elephants, sea creatures, whales, they do not have sweat glands, so that's why they love to roll around in the mud. Or the elephant is pouring, the sand on his skin, taking dust bath. So anyway, the saying what people says that you are sweating like a pig is totally wrong. You have to say you are sweating like a horse. And if you ever ridden horses, I'm sure you know that after finishing the ride, you have to dry off the horse. You have to remove the sweat. And it's really stinky, I have to say. I used to do that for a while. Okay, if you like tables, again, this is the sebaceous gland is compared with the sudoriferous sweat gland. So I'm comparing the two major sweat glands with each other. But again, you cannot miss it. If you see fat-looking gland around the hair follicle, that has to be sebaceous gland. If you see circles, the ocini look like circles. That's basically the coiled snake sweat gland. Okay, so now comes the cutaneous receptors. Okay, I just want to check the 10 till 5. Okay, so I have a question that this is going to be the next section, the mechanoreceptors. Can we have like a 5 minutes break?
Yes, please, doctor.
I think everybody needs a 5 minutes break, get a glass of water or have a snack or something. I will be right back. Good? Okay, I'm here. Okay, participants 68. Now that's out of the 400, that's not much. Okay. Let me know when we can start again. Okay, as I said, please let me know. How about we say we start at 8? No, not 8. What am I saying? 6.
Yeah, sure.
Okay, so just so that if somebody wants to whatever grab a snack or something, then we continue at 6 exactly. Okay, I'm back. Can you guys hear me?
Yes.
Okay, so we're gonna continue with the sensory receptors, or you can use many names here, depends on what you want to emphasize. You know, sensory receptor is a general name for sensation, but since we are doing the receptors in the skin, we're gonna say cutaneous and mechanoreceptors, because technically these stimuli, what they pick up from the outside world, are mechanical stimuli. If we, the specialized, the eye, ear, and the special receptors are all on your head and very complicated. These cutaneous mechanoreceptors is mainly sitting in your skin, and they are everywhere. Okay, so, and they can be in different layers of the skin. We can do classification of the stimulus. The stimulus can be, as I said, they are all mechanical. They can be harmful or not harmful, depends. Okay, light touch, stretch, vibration, move of the hair, and it could be, of course, harmful, pressure, harmful, hurting, piercing, blah, blah, blah. Classification of the receptors by structures. We have two groups that we're going to analyze. There are 3 receptors that have no connective tissue capsule. Then you have not 3 receptors that have connective capsule around it. And we're going to analyze it just in a second. What I would like you to remember, what these teeny tiny little organs can do. The major function of these teeny tiny organs are very simple fellows altogether, to compare to the ear or the eye. They are doing mechanotransduction. What does it mean? That mechanical stimulus, for example, touch or temperature, they sense it and they turn it into action potential. They are capable of making action potential from the stimulus. And I'm sure there's a lot of versions here, depends on what receptors you are looking at in detail, but most action potential means that some sodium channel opens up. Let's talk about the adaptation to a stimulus, that meaning how long you are capable of feeling, sensing of that sensation. And there are some receptors that are rapidly adopting, and that very quickly, you remember when you put on the watch and 5 minutes goes by and you don't feel the watch anymore. So your receptors adopted to that stimulus. So it can be rapid very quickly, or the adaptation can be also very slow. Receptors who has connective tissue capsule usually rapidly adapting. And the reason is, if you think a little bit just like common sense, that the connective tissue capsule is blocking the stimulus hitting the sensory nerve sitting in the middle of the capsule. Because just make sure that you understand, the receptor is not the capsule. The receptor is a sensory nerve within the capsule. You will see an image in a minute. And those receptors have that without capsule or no capsule, they are slowly adopting. So the capsule is not blocking the stimulus, so they can monitor the incoming stimulus for a longer period of time. So therefore, they are slowly adapting. Very important that the location of the receptors determine their function. If they are closer to the surface of the skin, then obviously they're going to be more sensitive to the stimuli. And those 3 receptors that have no connective tissue capsule, you can also imagine that even though slowly adopting, but they are actually more sensitive to the incoming stimuli. So these are kind of like the basics that I wanted you to understand. And these are the abbreviations that I'm using. Okay, so this is the famous guys because all of these sensory receptors are named after a scientist. So this is Mr. Meissner, this is Mr. Pacini, this is Mr. Ruffini, and this is Mr. Merkel. And they are associated, with the Meissner corpuscle, Pacinian, Ruffini corpuscle, and Merkel disc after this guy. Okay, so we're gonna go through all of these guys one by one. And there is one nerve or receptor that I didn't mention is the free nerve endings. So this is the most common, the most important receptor actually in our body, but it doesn't have any big name like the other ones. Okay, so I'm gonna start right now with the capsulated or the receptors with capsule. So the Meissner, the Pacinian, and the Ruffini corpuscle belongs to that. Okay, so I put them here in beautiful comparison so you can see them, that the Meissner is, the capsule looks like a coiled yarn, and made out of collagen, Schwann cell, and fibroblasts. The Pacinian corpuscle capsule looks like an onion in concentric layers, but again, it's made out of collagen, Schwann cell, and fibroblasts. And the Ruffini corpuscle capsule looks like elongated fusiform or spindle shape, right? Again, made out of collagen, Schwann, and fibroblasts. So these are the components of the capsule. So where is the sensory nerve? In the center. You see, it's just entering here, and in the Meissner corpuscle and the Pacinian corpuscle, all you see is one single unmyelinated sensory nerve ending that is not branching. And here in the Meissner, this nerve is actually spiraling. What about in the Pacinian corpuscle? In the Pacinian corpuscle, you can see it's just straight, one unmyelinated sensory nerve ending and no branching. You can see how as the sensory nerve comes in to the center of the capsule, you can see the Schwann cells and the node of Ranvier, but they kind of lose the Schwann cells. However, some of the remnant of the Schwann cell contribute to the corpuscle. Now let's look at the Ruffini corpuscle. Now this guy looks really different. The nerve enters, right? One unmyelinated sensory nerve ending, but once it goes into the capsule, it's branching like crazy. Lots of branching starts occurring left and right. Okay, so let's look at their function. So let's say the Meissner corpuscle is always in the papilla or the papillary dermis of thick skin. So therefore, if you think about it, it is very, very close to the outside world, to the reticle, to the epidermis, right? Very close to the epidermis. So therefore, it's gonna be very sensitive for light or surface touch. So the moment you have a slight touch, the Meissner corpuscle is going to pick it up in the thick skin. And it's going to be rapidly adapting because if you remember, we just decided that those receptors that has a connective tissue capsule, they don't monitor the incoming stimuli for a long time. So therefore, they are rapidly adapting. And now let's look at the Pacinian corpuscle in comparison. This guy is sitting in the deep reticular dermis of the thick skin. And it's interesting, we can also find this guy in pancreas. So let's look at, since it has a capsule, it's also rapidly adapting, but because it's located very deep in the reticular dermis, it's gonna be sensitive to strong, coarse, deep touch. So this guy is not waking up from a light touch or light vibration. This guy needs a very heavy vibration to wake up in the deep of the reticular dermis. also observe that both of them are actually located in the thick skin. So what does that mean? It means that the thick skin needs help because it's thick. So therefore it is less sensitive. So we have these extra receptors in the thick skin. So we feel the outside world, we are more sensitive to the outside world, also at the fingertips and our soul. So let's look at the third guy, okay, which is very different. This is an ubiquitous sitting all over the dermis, even though I have never seen it on a microscopic slide, and probably because it's very small. And so it's sitting in the skin, and it's also show up in MSK. I'm not sure you learned about the Golgi tendon organ or the joint capsule. In the joint capsule and the ligaments, where they actually monitoring the strength that goes through in the tendons and in the muscle. And these guys are, even though they have a capsule, they are slowly adapting. And I can explain to you with a common sense that the slow adaptation is directly related to the fact that it is not one single nerve that is picking up the sensation or stimulus from the outside world, but many little branches picking up. So therefore, it can monitor the stimuli for longer time. Therefore, it is slowly adapting. And this guy in the Golgi tendon organ is a stretch receptor. Okay. So I wanted to share with you that in 2021, these two guys got Nobel Prize for mechanotransduction research. Okay. So if you are interested in reading about them, here we go. Okay, so if you like tables, everything that I just did about the receptors that have capsule, the Meissner that is sitting in the papilla, the Pacini that is sitting in the reticulodermis, and the Ruffini that is everywhere, and MSK, then here is in the table everything summarized. Okay, so now I'm going to show you those receptors, guys, that are without capsule, so no capsule at all. And we're gonna cover three guys. The Merkel cell that we touched on lecture 1, but we're gonna talk more about it. Then at the hair follicle, you actually have two kinds of receptors, free nerve endings that are actually hugging the hair follicle, and this bunch of Merkel cells hugging the hair follicle that people usually call Merkel touch dome. And what I mentioned to you earlier, the free nerve endings that are really the number one receptors, and it's called free because they do not have capsule. Okay, so let's look at them one by one. The epidermis touch receptor, observe, this is actually a cell. Okay, it is not a receptor organ. All of the other receptors that we covered here, they are not cells. They are multiple cells. They are Schwann cells, fibroblast, collagen, a sensory, nerve ending, neuron. These guys are relatively complex, but this guy is really just one cell sitting in the stratum basale of the epidermis. And it is located in all skin and even in oral mucosa. This guy, of course, cannot have a capsule in the stratum basale. So if he has no capsule, right, then what did we learn? That slowly adapting. It can monitor the incoming stimuli for a longer time. Therefore, it's slowly adapting. But it's very close. This is the receptor that is the closest to the outside world. Well, free endings are pretty close too if they enter the epidermis. So therefore, they wake up from gentle touch and vibrations. So this is everywhere, the Merkel cells. Now the hair follicle, what I just described, the free nerve endings and the Merkel dome, obviously is only at the hair follicle. And because they do not have, again, capsule around them, that's why slowly adopting hair. And remember, this is the reason we can sense if somebody is pulling our hair. And finally, let's analyze the main mechanoreceptor that located all over the body, not only in the skin, epidermis, dermis, and of the all kinds of skin, and they can pick up pain, which we call nociceptor. They can be temperature receptor, touch receptor, and again, they're gonna slowly adopting because they don't have capsule. Okay. So here we go. So this is, I borrowed this couple of slides from lecture 1 because I didn't want to share back and forth. So this is the detailed description of the Merkel cell, which is also called low level clear cells. And I said it's another because you remember the melanocyte is also low-level clear cell. Low-level means that it's in the stratum basale, and it's clear that when you look in the microscope, you're gonna see white cytoplasma. It looks like an eye looking at you. And yeah, exactly, hard to distinguish from melanocyte. And yes, and yes, we just did that. Free nerve ending, Merkel cell, and the hair follicle. Very good. So let's move on. And this is the structure, function, and location of the Merkel cell. We just did that, the location. Very good. Embryology. Neurolike crest origin or epithelial origin. People argue about it. It's not very big, around 10 micron. No capsule, as we did that. Only one cell. It's not an organ like the other guys. One unicellular general sensory receptor. Dendritic cell, like all of these epidermal cells. The nucleus is lobe that you can't really see on light microscope. And they have desmosome with keratinocytes. Now this is where I want to ask, you remember melanocytes next door do not have desmosomes, but these guys have desmosomes. So I suggest to you, again, just like common sense thinking, is that these cells are monitoring and they are very sensitive to the outside vibrations and the outside touch. One more thing I wanted to make sure that you understand that these, if I go back here, you see, the Meissner corpuscle is wakes up from little vibration because it's very close to the surface. So this is also true to the Merkel cell, very close to the surface. So very light touch and very little vibration, very slow vibration can activate it, as opposed to the deep reticular dermis where the Pacinian corpuscle is. You need a really big stimulus and a very big vibration to wake this guy up. So here we go. So because this guy is so sensitive to everything, I think it needs to be anchored so it doesn't move from anything other than the incoming touch stimulus. Okay, so then this Merkel cell is going to synapse with a sensory nerve, and this is what we call Merkel corpuscle or Merkel cell neuroid complex. Okay, where is it located? In the epidermis, touch receptor, stratum basale, thick skin, thin skin, mucous membrane. So basically everywhere. Okay, this final is about just the function. It's a general sensory receptor for gentle touch. And you remember location, very close to the surface. This is the closest because it's in the epidermis, so therefore very sensitive, and it doesn't have a capsule, so it's not getting tired, so it's slowly adopting mechanistic receptor for gentle touch. So wakes up from fine and gentle touch, wakes up from low vibration, monitors long pressure because it doesn't get tired, and it's also involved in finger precision, recognizing shapes, edges, and textures. So when you put your fingers into your pocket and you recognize that, oh, this is my wallet, this cell helps you to identify these structures. Okay, and here is the synaptic vesicles, and here's the sensory nerve that is picking up the sensation, because you remember the Merkel cell creates the action potential in the sensory nerve that is directly heading to the postcentral gyrus. And if you like tables, here we go. Everything is summarized in this table about the receptors that have no capsule. Okay, this is a summary table about desmosome. We have desmosome between keratinocytes. We have no desmosomes for the Langerhans because they are policemen going all around. Okay, melanocytes have no desmosome, and if you remember, what I said was that if the keratinocytes is biting off a piece from the melanocytes, then desmosome shouldn't really be there, blocking this process. The Merkel cells has to be anchored because if they are continuously moving, they're gonna monitor false stimuli, not a touch from the outside world. And sebocytes also have desmosome. Now, one more thing I wanted to show you, the classification of the based on thin skin and the thick skin. So thin skin have free nerve endings. Of course, the hair have the nerve plexus, which is technically another type of free nerve endings. We have the Merkel cell in the stratum basale, and we have the Ruffini. So these are all in the thin skin. And I put it here for you, reminder, the thick skin needs help. It's too thick, so we need to make sure that it is sensing the stimuli from the outside world. So we're gonna add on the top of the free nerve endings and the Merkel cells and the Ruffini, the Meissner corpuscle and the Pacinian corpuscle are gonna be the extra help in our thick skin. And you remember thick skin have no hair, so that's why the hair nerve plexus is not located here. So this is a summary slide for you about the this is the pink Meissner corpuscle sitting in the papillary dermis. This is the orange Meissner corpuscle coming from the finger slide, sitting again the papillary dermis. And we have three kinds of Pacinian corpuscle slides, a pink one, a bluish one also from the orange slide, and we have Pacinian corpuscle in the pancreas that you can also observe very nicely here. So this is, you can see the parallel organization of the capsule on both. And here you can even see the sensory nerve. And you can see that some of these Pacinian corpuscle in the pink and the blue is kind of, the shape is a little bit distorted. Maybe during the processing they got squished or something, but you can see that the center is where the sensory neuron is located, and here and here. But the Pacinian corpuscle in the pancreas has beautiful, perfect oval onion shape. You can see very nicely the parallel organization of the capsule. Okay, like an onion, layers of onions, layers of onions, layers of onion. Now on this, guys, of course, the sensory nerve is in the center, so we can't really see it. The capsule covers it up. And again, these guys are going to be very sensitive because they are close to the surface. These guys are not that sensitive because they are deep in the reticular dermis. And so this is kind of repeat here for you on the top. Now these slides are a couple of interesting application. What structures of the skin is involved in these cosmetic procedures? The Botox injections, probably you know, paralyze the skeletal muscle under the hypodermis. In theory, this is going to relax the muscle. Therefore, the skin is relaxing, and therefore less wrinkles show up. The dermal fillers have extra collagen, hyaluronic acid, and lipid that they inject into the skin here or the lips. Then I don't know if you ever heard about the PRP, platelet-rich plasma, where they, what is really nice that they take the blood, they centrifuge it down, and they take the platelets, your very own platelets, and they inject it back under the skin. And it has a rejuvenating effect. Actually, if you want to have any kind of rejuvenation, you really want to wake up the fibroblasts in the dermis. So this platelet growth factors is, in theory, renew or wakes up the fibroblasts to create more collagen, so your skin looks more tight and more young. Okay, face peeling scrubs. You can find a lot of different scrubs. They put little rocks in it, and then it removes the surface of the stratum corneum. So your skin is going to start looking more shiny, more glowing. But you have to be careful here, because if you do that, you really should put a lot of sunblock on you, because you can hurt the skin. The laser skin surfacing, I don't know if you ever heard about it. The laser, this instrument, removes the epidermis layer by layer. But the same idea, stimulate the fibroblast, and also brand new epidermis grows up, which is new, glowing, smoother, and firmer. Okay, the last slide here is showing you the blister that we talked about the other day. Here's the image for you. This is the epidermis that is dying because the fluid under it blocks the oxygen delivery coming from the capillaries of the dermis. Okay, so I think we covered this, and we talked about it, that the hemidesmosome broke in half, so broke off, right? That's what and mechanical tight shoes can create this. There's many other reasons, but this is what we covered. The freckles are overproduction of melanocytes in different spots. And when the skin is peeling off like big pieces, the desmosomes are still there. Okay, it doesn't look like dandruff. And this is showing you the tattoo, but the pigments are entered into the, look at this on the left side, into the deep dermis. So this is the tattoo you can't really get rid of. If you want to get rid of a tattoo, you want to go not deep in the dermis, but only in the epidermis, so eventually it's going to fall off. Okay, couple of clinical situation that you're going to learn more about it from the other people. But lipid soluble drug can be delivered by skin patch, like nicotine, if somebody's suffering from cigarette withdrawal syndromes, steroid hormones, seasickness drugs. Albinism is when this is a genetic hereditary disease where the absence of an enzyme tyrosine is absence of an enzyme, basically melanin is not synthesized. So it's pretty dangerous if there is no pigmentation of the skin. So again, you have to hide from the sun. The another one is very important, that whenever a patient is going to walk into the clinic, and immediately you could see that the skin is healthy colored, healthy texture, healthy look, and or you can see the color is blue or yellow or pink, or the hydration, the person is hydrated, not hydrated, the nails are breaking, not breaking, the hair is healthy. But basically, the skin really helps immediately give you a clinical kind of impression about the person. Obesity, when we have too much hypodermis, right? But unfortunately, the fat accumulates not only under the skin, but inside the organs as well. Like, for example, in the heart, where the epicardium is. Now, the next couple of guys are all related to cancers caused by often sun's UV radiation, like basal cell carcinoma. And it's called like that because it looks like the basal cells of the epidermis, but this is not very dangerous. It's a slow growing tumor, usually does not cause spreading or metastasis. Now, the squamous cell carcinoma is more vicious, is a small painless nodule with inflammation on the skin, and it can show up in all layers of the epidermis. The malignant melanoma is most dangerous, very important to detect early, and early surgery. And these are the ABCD rules of melanoma: asymmetrical shape, border of the tumor irregular, C, color is multiple, D, more than 6 millimeter size. And the Merkel cell carcinoma, that teeny tiny cell in the stratum basale, the epidermis, can cause really vicious tumor as well. Rare, very aggressive tumor. One can see painless lump on a sun-exposed skin, grows very quickly, and metastasis occurs through lymphatic vessels. Now, there's a second cause of Merkel cell polyoma virus. It's a human oncovirus, and if somebody gets infected by this virus, can develop Merkel cell carcinoma. Okay, so the lecture is over, and now it's 6:30. So I just wanted to say that there's one more thing I would like you to do. What I would like to do is stop sharing this. Anyway, if anybody has question, that's one thing. The second one is I want to take again a 5 minutes break, and then I'm going to show you the Olivia slides. Okay?
Okay.
And then it's probably going to be able to finish by 7 o'clock.
Okay, that's great.
So 5 minutes break. It's what is it right now? 32. Okay.